Stay a while and listen.
Mitochondria are the fuel refineries of our cells, using food and oxygen to create energy that powers almost every cellular function. Here, mitochondria are localized near contractile units within the muscle fibers, supplying the energy for muscular work. Within the mitochondria, a complex dance of molecules harvests the chemical energy from sugars, fats, and other nutrients. While generating power, mitochondria accidentally produce toxic molecules known as reactive oxygen species. Occasionally, these toxins react with the mitochondria's own DNA, damaging genes critical to the mitochondria's functioning. This causes the mitochondrion to cease to function normally, impairing cell function and becoming toxic to the cell it's meant to fuel. The dysfunction of those cells then contributes to the diseases and disabilities that plague those specific tissues with age. Mitochondrial mutations are especially important in the neurological disorder Parkinson's disease and the age-related loss of muscle strength and quality that even active people suffer. But what if there was a backup to the mitochondrial DNA? Using gene therapy, mitochondrial genes could be delivered to cell nuclei, where genes are kept safely away from the more violent cellular processes. When there's a mutation in the mitochondria, replacement proteins would be supplied by the backup copies of the genes. Fortunately, our cells are already equipped to do this. Only a few critical genes are housed in the mitochondria. 99% of all mitochondrial proteins are actually already delivered this way, from genes that are naturally housed in the nucleus. The cell will simply use this same machinery to deliver the few missing proteins that the backup genes encode. With all the missing proteins now replaced by backup versions, mutant mitochondria would still function normally and would no longer be toxic to their host cells. These cells could then return to health and normal function.